Most people understand the basics of inheritance. You get one set of genes from one parent's egg and another set of genes from another parent's sperm, and the two gametes join and form a zygote that eventually turns into you. A perfect mix of both of your parents' genetic makeup. But that's not quite how inheritance works. In fact, we are quickly learning that inheritance is much, much messier, especially when it comes to gene expression. One set of biological mechanisms that can control gene expression is epigenetic traits, with epi meaning on top of and genetics meaning genetics. The term epigenetic refers to the traits that change the way your genes are expressed without physically changing any of the nucleotide bases of the DNA. Think of epigenetic mechanisms as the genetic managers of the office. They're the mechanisms above your genes, controlling when and under what context they can be read and do their jobs. There are many mechanisms of epigenetics, many of which are inheritable, and one of these heritable mechanisms is called genomic imprinting. Genomic imprinting occurs when one of the genes we inherit, either from the sperm or the egg, is silenced. Silencing usually occurs through DNA methylation, where a chemical tag near a region of DNA tells that section of DNA to coil up even tighter around its protein in its folded up state, making it unreadable and, in this state, non-functional. Usually, these chemical tags are reset during sperm and egg formation, but sometimes they're not, and then they get passed down to the offspring. Genomic imprinting occurs when these chemical tags cause only one parent's gene to be expressed, either the X gene or the sperm's gene, and the other copy to be silent. In mice, we currently know of about 150 genes that tend to undergo imprinting, but we're likely to find more as we continue to research. And because imprinting patterns appear to be relatively similar across mammals, studying mouse models is a great way to understand more about the human genome. But why do we even care about this process? I mean, what happens if a relatively small proportion of genes gets turned off occasionally? I mean, we get two sets of genes anyway, so does it matter if one set of genes gets turned off in some places? It matters a lot. In fact, imprinting is essential for important processes like normal fetal growth and general development. For example, the typically paternally expressed IGF2 gene is a positive growth regulator. And overexpression of the paternal copy can lead to fetal overgrowth, while underexpression leads to growth restriction. Also, related to embryonic growth is the typically maternally expressed gene GRB10. In mice, when this maternally expressed gene is shut off, the embryos end up underdeveloped. And when the imprinting control regions are shut off, causing both alleles to be expressed, the embryos also end up underdeveloped, demonstrating the importance of imprinting as a control mechanism. Imprinted genes can also affect traits more visible later in life, like behavior, or even cause a wide array of disorders. Take the gene UBE3A, which has both parent genes expressed in most tissues, but in brain tissues, only the maternal gene is supposed to be expressed. This is actually a pattern of expression found in multiple cases, where the direction of imprinting, whether the paternal or maternal gene is expressed, sometimes switching depending on the tissue type or the life stage in which the gene is being expressed. When the UBE3A maternal gene is deficient, a disorder called Angelman syndrome occurs. Another gene that only demonstrates imprinting patterns in brain tissue is the PEG3 gene, which regulates neuron cell death. PEG3 is paternally expressed, so deleting the paternal copy results in increased brain cell death in neonatal specimens. PEG3 also affects behavior. Without the paternal copy, the maternal abilities of female mice are affected, causing deficiencies in breastfeeding and nest-building behaviors. And in baby mice, we observe them being worse at breastfeeding, with the result in suckling behavior. Back to the familiar GRB10 gene, in mice, deleting the paternal allele, which is expressed in certain neurons, is associated with hyperaggression and increased social dominance. In summary, imprinting is very important, and there are a wide variety of disorders related to the misexpression of imprinted genes, with misexpression related to either the over or underexpression of a gene that typically undergoes imprinting. These disorders range from forms of diabetes to syndromes that increase your risk for certain cancers or behavioral and developmental abnormalities. But why? As in, how did this mechanism of control come about? I mean, it affects many traits that conceivably have a profound effect on fitness. So how did the phenomenon of genetic imprinting become so powerful? Well, there are multiple theories about why imprinting was selected for. Here, we'll just be looking at one. One theory, sexual antagonism theory, argues that imprinting is a result of natural selection treating males and females differently for a given trait. To better explain this point, let's say we have a population of dragons. For dragons, scale color is a heritable trait, and these dragons can be a wide variety of colors, but males have a better chance of living to reproductive age and passing on their genes if they're blue, and females have a better chance if they're yellow. So natural selection will treat males and females differently. This difference in selection results in the males and females of a population evolving to have different allele frequencies. So in our dragon example, ideally, there will eventually be a majority of blue male dragons and a majority of yellow female dragons. Suppose a blue father and a yellow mother go to have a baby dragon. In that case, imprinting can be highly beneficial because now the offspring will likely carry two sets of genetic information, and imprinting will allow it to potentially express the blue trait if it's a boy dragon or the yellow trait if it's a girl dragon, maintaining its selective advantage, rather than being blue whether or not it's a boy or a girl dragon, or turning green or something. Under this proposed evolutionary mechanism, the main requirement for imprinting to become common in a population is some kind of sex-specific selection on a gene. And for a species with two sexes, this kind of selection is not rare, allowing this theory to apply broadly to many species. So, what have we learned? Inheritance is weird, and DNA itself is far from the only factor at play when it comes to how genes are expressed as traits. One of these factors at play is called genomic imprinting, which leads to only one of the parent's genes being expressed and the other being silenced. Additionally, these changes are not consistent throughout the body, with imprinting occurring differently at different stages of life and development and in different types of tissues. These changes are really widespread, affecting traits related to everything from growth, development, and social and parenting behaviors. Finally, we still aren't completely sure how or why this mechanism evolves, but one theory is sexual antagonism theory, which relies on natural selection affecting males and females of a population differently. I hope you enjoyed this video about genomic imprinting and continue to learn more about the strange workings of inheritance that make us who we are. I know I will.